Welcome students. I think today I have taken a drug which we are doing in the lab, especially one batch have completed yesterday, ephedra, and I am very happy that the students are now well trained and their sections were amazing, and still more you have to improve. And I have just taken some of the section which have taken by the students also. Today I have taken as a photograph to show even in this class. So it will be an interesting for the students to make even the next practicals also. Students will be interested to do more and more, and your sections are very amazing, some of them, and still you can improve. And you see, this is ephedra. Today we have taken ephedra, and a batch have finished that ephedra. Many other batches they will start doing in the coming week. So in that you can have a good experience, and also you can share what all the things which you have done from the A batch students, and you can record properly in your observation and also in your record. See, this is the dried stem. Actually we are using the aerial part stem, and it has more medicinal value, especially when I was asking with the student in the A batch, they were telling about adrenergic drugs, and they were sympathomimetic drugs. More detailedly they were explaining in technical terms. It shows that students are in a good background, but you need some reading for you to write the exam. So now you see, this one is stem, aerial part, dried stem, and which we have done section today. Most of the students have successfully done this section for the stem, and I told them, especially, and also for you also, you all students, you can have a reference about stem parts, because we have seen bark, a leaf, flower, then comes to the stem we have seen, and next we will go for umbelliferaceae fruits, because only that much we can do, because we have to go to TLC, paper chromatography, and some identification also is there still for your experiments. So we have to go a long way. So that's why we are just giving one drug in one area, not one area, in one parts of the plant. And here you can see in ephedra, the plant, as I told, it has a flowering time. You can see it's a reddish flower, beautiful flower, can be easily identified as shown in the picture. And you can see it's a synonym is Ma Hong. Actually it is a Chinese synonym, Chinese, more geographically more identified in China, and the synonym is Ma Hong. And you can see botanical source, it has many species, that is ephedra consists of the dried aerial part of ephedra gerardiana. Ephedra gerardiana is the species which we are having in our lab, and we are performing with that sample. And there is other species also, but you can see this gerardiana is identified by the scientist Wall. That's why ephedra gerardiana wall. Ephedra sinica Steph, that is a scientist name, is sinica, another species, and Ephedra euchystenia, or you can say Euchystenia. This is the scientist which identified is Bungay. And Ephedra nibrodensis, that scientist which identified is Tinio. But we have in our lab Ephedra gerardiana. The wall is the scientist who have identified, so we remember the wall after the species. The other species are also available, which is also having the active principle of ephedrine and norephedrine. Epiephedrine is also present in most of the species of Ephedra. Family Ephedraceae. Family Ephedraceae. Geographical source, as I told, China is found in China mainly. That's why we are giving the synonym Ma Hong. In India, it's available Nepal, Turkey, Pakistan, and Bhutan. These are all the geographical sources and distribution of this plant we have taken. And now you can see in the plant which we have taken in our lab and performed, so that species is gerardiana, Ephedra gerardiana. This is the plant, and when we want to say about this morphological character, better to draw the diagram and give the parts also. See, nodes and all, you can just mark it very clearly. It's very easy for the students to mark. Whenever you draw a diagram, you should label them. Labeling is very important, especially a pharmacist when you want to formulate or when you want to dispense any drugs. Formulating a drug is more than formulating. Labeling and distribution, labeling is very important for any type of drugs, a finished product. Same thing, when you draw a diagram of a plant or a part, whichever you draw, it is necessary for you to label them properly. So that's very important. So what is it? It consists of this gerardiana is a species which we have taken for today's study, which is present in our lab. It consists of cylindrical woody stem, that is grey or greenish in color. What we had is grey in color because dried. Nodes, you can see the nodes will be there. A stem, if you take a stem, it will be having a nodes, like sugar cane is also a stem which we are using. It has nodes, small short nodes. So there will be nodes even for this stem. There will be internodes and scale of leaves in the nodes. Terminal buds also present. Some small buds may be present in the near to the nodes in the stem. And the distance between the internodes is 3 to 4 centimeter, and the node bear scaly leaves also. You can see the picture, there is some scaly leaves also present in the nodes of the stem. They are bitter in taste. So this is about the morphological characters. You can extend little more about the color, flavor, that which is not added here. You can just write even in the powder microscopy. You can go for reference in the practical books and you can write about when powder microscopy, organoleptic characters are very important. When we talk about microscopy, I have just jumped to the microscopy. I did not mention much about the morphology character, that you can add a little more what you observe, because the product is available lively in the lab. So you can identify, and also you can refer some of the practical pharmacognosy books, and you can elaborate your description in your record notebook. See, in the microscopical character, our students have taken most of them beautiful section, in which you have seen these sections, especially they have seen the curve, that is ridges. Actually, we did not discuss in the lab about these ridges, but the ridges have major role even in the next experiment when you are going for a fruit section, because when you fennel or coriander, the primary ridges and secondary ridges plays a major role. Here also you can see ridges means the curved surface of the outer surface, you can see like a heap, that is the ridges we are telling. And down, when it is curved one, that is in between two ridges they have mentioned here. But when we come to coriander, fennel, we used to say primary ridges, secondary ridges, like used to say, that is the in between two primary ridges, we used to say secondary ridges. Here also we have mentioned that ridges here very clearly. In the ridges you can see sometimes stomata. That's what we want, that's where I want to explain you, which we can see some stomata also may be present in the stem part. Understand? That's what we can see here in this plant ephedra. So the transverse section of the stem shows epidermis composed of thick walled quadrectangle, that is quadrangled, or you can say quadrangular cells. Quadrangular cells. It is not triangular, it is quadrangular. Four sides quadrangular cells you can see, and our students have taken very clearly the quadrangular cells in their section. I will show the photographs which I have taken also. Covered with a thick cuticle. The outer area will be covered with a thick cuticle. And you can see, as I told, ridges, you can see shringan, shangan, you can say shangan, not shringan, shankan stomata. Shankan stomata, you should mention that shankan stomata, you should know shankan stomata, specific stomata, parasitic stomata, animocytic stomata, like the shankan stomata, which we can see in the stem specifically, is present in between many vertical ridges. Vertical ridges, that means you can see the picture which I shown stomata. It is the ridge which is curved part, the down part. The ridges means that the heap part. So two heap part, a shallow curved part, that is the place which we were meaning that stomata is present. So now you see the parts which is mentioned, remember, very easy to draw and to mention: epidermis, stomata which is present in between the two ridges, and hypodermal fibers can also be seen, pericyclic fibers, and the xylem can be seen, which is lignified, and certain distributed fibers can also be seen near, entering near to the pith area. That's what the diagram which I want to show you. Now we'll come to the slide section which is taken by our student in yesterday's lab by Ranjita. I do remember the student, she took a very thin section, amazing section. You see that the same section I have just taken a photograph by means of my electron microscope, which I connected with the Android, and I took the photograph. You see how fantastic. When it is a thin section, you can see almost all the parts. You can see the ridges also, you can see the fibers, you can see the pith with darker spots, and also you can see the xylem and phloem, which the shape is so amazing shape. You can see the cortex region where, which just now we have shown here, you see, here which we have shown the xylem and phloem, that is the quadrangular cells also can be seen here very clearly, and fibers. And see that the ridges, see how she has taken a very good section, amazing section, and it was impressed me, and every students, almost all of them, they did very well by means of their own skill. It's not our, we just made a line, you made a big diagram. It was so amazing and appreciable work. And see here, what is the section by the her section? We are just pointing out. Papillae are present in the ridges. Papillae. What is that papillae in the ridges? I told ridges means the curved area, the hip area of the outermost surface, epidermis or cuticle below. Below the ridges, groups of non-lignified, as I told, there will not be any staining may not be occur, because fluoroglucinol cannot react with lignin because they don't have lignin. Unlignified hypodermal fibers are present. I will show you even some of the students who have taken the section. I have focused in the microscope and taken a photograph of that also. I have incorporated in this lecture so that you can see how your reality in the experiments. When you do some lab work and reality, if it is shown to you, you will never forget. That's the main aim for me to take a photograph and to show in my lecture. So you see, unlignified hypodermal fibers 9 to 20 per groups are present in the ridges, below the ridges. And cortex region, that is the cortex region which we are telling, cortex will be there in the stem. We know that. The cortex region composed of chlorenchyma, that means chlorenchyma. Chlorenchyma, we are telling that the chlorine, that is means, for example, some greenish tinge may be there. Chlorophyll will be there. There is a chlorenchyma also there, with outer zone of radially elongated cells. Radially elongated cells. You can observe their cell shape also. Even in microscope, enlarged in 45x, we have seen, and I have taken a photograph also. This is done by our student in the A batch. An inner zone of spongy parenchyma. Inner zone, you can see the spongy parenchyma. Cortex also contain few isolated fibers. See, there's few isolated fibers, pink color here and there, dot by dot, are groups of fibers. 2 to 6 per groups. 2 to 6 per groups. Very amazing section which we can find out very clearly. Which are lignified. They are lignified. That's why it is pink in color, which can react with the lignin which is present in the cell, the fluoroglucinol which you have added. And this is another section which is taken by our student. I hope it is by the next student, behind. I remember, I think it is Drishya. It was little thick. That's why you can see the pinkish color, but the cells are not clear. Only the pith can be seen. But we were unable to see the xylem and phloem cells differentiated in the distributed fibers. So this is little thick, but anyhow it's also, it's not bad, it's not excellent, but it is good. So just that I want to show you. And there are some other students also, they showed me, even I do remember some 2-3 students, they showed me staining was poor, but the section was so thin. But if they have stained properly, if they have waited for some certain times, at least 5 minutes, after adding the concentrated hydrochloric acid and fluorogrocinol, even if it is warming also can be done. I have told, but our, I think, lab was not, everybody, there is some, the technicians who is coming is changed always. That's why we insisted you have to switch on the, especially you have to switch on mainly the water bath when you enter the lab. So that's why I was telling that if you warm, staining can be more feasible, more successful. Anyhow, we will do it, but anyhow, this is also got very good result, and it was amazing work by the students. And this is the unlignified fibers which is present below the ridges. How clearly you can see, without any staining, without any, that means it is unlignified. So fantastically, you can count itself. How many numbers of fibers can also, how they said it is 6 to 9, that also so amazing sections taken by the student, just by the A batch student. This is, I remember, this is by Maria James, and also done by, I think, Sona Thomas. Some of the, most of the students, many of the photographs I took, I did not mention, I cannot show all, but I took most of them. It was amazing section today, an amazing lab yesterday, which I had with A batch student. They did very, very fantastic work. So again I am repeating about that is papillate are present in the ridges, the curved part, ridges, below the ridges. That means ridges, I told, ridges means a curved and then like this, like this. So the heap area is ridges, below the ridges, cuticle, epidermis, and then comes unlignified fibers, non-lignified hypodermal fibers. 9 to 20 per group will be there. 9 to 20. We have numbers are 9. So I was amazing, 9, 10 fibers are there. Very clear picture. And this is the pitta. I think this is also done by a student, and she was taken, I was telling about why it looks like rock stones. The pith was so, it's a darker browny spots will be seen. That is also, you can see brown spots, cells will be seen in pith area. And we focused in the microscope and we took this photograph. I do remember the name of the student. Let me, we will, I think she knows. I told her, even while observing in the microscope, I told it looks like stones. So it was so amazing section which was observed, and I took that photograph. The pith is large and round cells. You can see round cells. How beautiful, how amazing. The section was showing the round shape, containing dark brown mucilaginous substance. The dark brown spots, you can mark it. Even in the diagram book even I have not marked, but you can mark that the dark spots which is present in the pith area, they are dark brown mucilaginous substance in the pigment cells. In pigment cells. And this is the diagram which we have drawn. You can see that a small description continuation. Pericycle fibers is composed of groups of lignified fibers outside the phloem region. Vascular bundles are 6 to 10 numbers, which we can see radially arranged in the cortex and composed of phloem and xylem. That's why phloem and xylem, then only we will say it is vascular bundle. So you can see the picture, so amazing. First one, epidermis, before that cuticle will be there, and the yellowish part which I am showing, that is the unlignified fibers which we have seen in the enlarged one also, our own section which I just now showed, without any staining, without any pinkish color, because it doesn't have lignin. And lignified fibers distribution also, we have seen certain spots in the cortex region of fibers, lignified fibers, and pericyclic fibers, as shown the arrow, pericyclic fibers. Phloem, xylem, you can see in the radially it can be seen. That's what we mentioned little before. And see here, pith area. So amazing pith cells can be seen, which contain dark brownish, which is, you can see, I am showing that dark brownish. See, there are many spots are there, which we have observed even when we did the section. Chemical constituents. Ephedra contains alkaloids, ephedrine, water soluble salts of an alkaloid, pseudoephedrine, among of ephedrine is also present, nor pseudoephedrine, an analog of ephedrine, is also present. The leaves and the stems of ephedra also contain many potentially active compounds such as tannins, saponins, flavone, and volatile oils. Ephedrine and pseudoephedrine, the structure which I have shown here is a simple structure. They both are same, only they are having an optical isomerism. The structure is not having any difference. Only thing is the CH, OH and a benzene this side, and this side you can see CH2, NH, CH3. They have different substituent, which is an chiral or chiral carbon atom. So when OH is below the plane, it is pseudoephedrine, and when OH is above the plane, ephedrine. That's what which is shown in this picture very clearly. Don't get confused. There is no change in the structure. They both are same. Benzene attached with CH, OH, CH, N, CH3, and one CH3 is attached. This is the molecular formula is same. Only the configuration is different. They are having optical isomers. Chemical test which we have performed in the lab, and most of the students, they got that purple color. Some of them they got only blue color, purple color is persist for some time and it disappeared. To the drug is 10 milligram in water, a small pinch, 1 ml, very little you have to add, 1 ml of dilute hydrochloric acid, 0.2 ml of copper sulfate solution, 0.1 ml of sodium hydroxide solution, 2 ml are added. The liquid turns violet on adding solvent 2 ml ether and shaking vigorously. The ether layer which is separated and it shows, which turns purple, and the aqueous layer becomes blue. So it is also performed by the student and they have observed. So it will be easy for you to remember what we did in the lab. If they ask any question, I hope my student will not miss anything to write. Uses: ephedrine is anti-allergic or anti-allergenic, or you can say antihistaminic, anti-asthmatic, antispasmodic, pain, it can relieve abdominal pains, antispasmodic, decongestant, if there is decongestant, the blocking in the nose, decongestant, cough suppressants, cough suppressant, if there is cough can also be suppressed by means of using this ephedra, stimulant and vasoconstrictor because of its presence of ephedrine. As most of the students have told about, it is an sympathomimetic and adrenergic drug, they said. And pseudoephedrine is also a decongestant, cough suppressant, and pseudoephedrine is peripheral vasodilator used in the treatment of angina. Angina pectoris. Angina, it can be used for as an peripheral vasodilator. That's why it can be used for angina. As a whole, it is decongestant. It opens sinuses, increases sweating, dilates bronchioles, and the asthmatic use, and it is diuretic, increases urine output, CNS stimulant, rises blood pressure in some cases, alleviates, not elevates, alleviates, that means reduces aches, that means pains, and rheumatic pains, rheumatism, alleviates rheumatism, alleviates hay fever, reduces hay fever, cold, etc. So it has an amazing category of uses which you can clearly mention in this drug. Thank you for being with me for the small class, and we'll have the discussion now.